what am I doing here? For those of you who don't know me, my name is Philip Buchholz. I have a PhD in biochemistry and molecular biology. I'm a cancer gene jock. Basically, I do cancer genomics research at the University of South Carolina. And what that means is that I'm kind of an expert on all the ways that the human genome can get futzed with during your lifetime and which of those things cause cancer and which ones don't. Okay. So technically, that means that I'm very, very skilled in the art of DNA sequencing. Okay, I can figure out the sequence of things that I didn't know what I was looking for. And I'm also pretty good, when I say I mean the people in my laboratory. You're not going to hear their names, but there's a group of people that do this excellent work. We're really good at detecting foreign pieces of DNA in places where they're not supposed to be, even if they're real low levels. And we used those skills during the pandemic we invented the COVID test that many of you did a spit test. Okay, that came out of my lab because we were really good at that kind of stuff. And so I've earned a fair amount of respect in the state of South Carolina and in this body because we did a ton of COVID testing in the middle of the night when people were afraid, and we told them, no, you don't have COVID in your home, or yes, you do. So my qualifications to comment on this are both technical and kind of relational in the state of South Carolina. Pandemic. And it was the best of sucky options that we had. And I still believe that it was deployed mostly in good faith, but there were a lot of shortcuts taken because the house was on fire, and we could do a better job next time. But I'm also, my philosophical bent here is, I'm sure many of you have heard of Occam's razor, right? Choose the simplest of explanations. Well, there's another one called Hanlon's razor, which is never attribute malice to that which can be better explained by incompetence. And so I'm trying to be gracious here in many circumstances. There could be malice underneath, but I'm trying to see just incompetence to be gracious. So the Pfizer vaccine is contaminated with plasmid DNA. It's not just mRNA. It's got bits of DNA in it. This DNA is the DNA vector that was used as the template for the in vitro transcription reaction when they made the mRNA. I know this is true because I sequenced it in my own lab. The vials of Pfizer vaccine that were given out here in Columbia, one of my colleagues was in charge of that vaccination program in the College of Pharmacy, and for reasons that I still don't understand, he kept every single vial. So he had a whole freezer full of the empty vials. Well, the empty vials have a little tiny bit in the bottom of them. He gave them all to me, and I looked at them. We had 2 batches that were given out here in Columbia, and I checked these 2 batches, and I checked them by sequencing. And I sequenced all the DNA that was in the vaccine, and I can see what's in there. And it's surprising that there's any DNA in there, and you can kind of work out what it is and how it got there, and I'm kind of alarmed about the possible consequences of this, both in terms of human health and biology, but you should be alarmed about the regulatory process that allowed it to get there. So this DNA, in my view, it could be causing some of the rare but serious side effects like death from cardiac arrest. There's a lot of cases now of people having suspicious death after vaccine. It's hard to prove what caused it. It's just, temporally associated. And this DNA is a plausible mechanism. Okay. This DNA can and likely will integrate into the genomic DNA of cells that got transfected with the vaccine mix. This is just the way it works. We do this in the lab all the time. We take pieces of DNA, we mix them up with a lipid complex like the Pfizer vaccine is in. We pour it onto cells, and a lot of it gets into the cells, and a lot of it gets into the DNA of those cells, and it becomes a permanent fixture of the cell. It's not just a temporary thing. It is in that cell and all of its progeny from now on forevermore. Amen. So that's why I'm kind of alarmed about this DNA being in the vaccine. It's different from RNA because it can be permanent. This is a real hazard for genome modification of long-lived somatic cells, like stem cells, and it could cause, theoretically, this is all a theoretical concern, but it's pretty reasonable based on solid molecular biology, that it could cause a sustained autoimmune attack toward that tissue. It's also a very real theoretical risk of future cancer in some people. Depending on where in the genome this foreign piece of DNA lands, it can interrupt a tumor suppressor or activate an oncogene. I think it'll be rare, but I think the risk is not zero, and it may be high enough that we ought to figure out if this is happening or not. And again, the autoimmunity thing is not my wheelhouse. I'm not an immunologist, but the cancer risk is. That's my bag. I know this is a thing, and it is a possibility. About 200 billion pieces of this plasmid DNA in each dose of the vaccine, and it's encapsulated in this lipid nanoparticle, so it's ready to be delivered inside the cell. Okay? This is a bad idea. My conclusions from this, we should check a bunch of people. My conclusions from this, or I should learn how to run PowerPoint. We should check a bunch of vaccinated people getting tissue samples, especially if we focus on harmed people, but that's not necessary. We could also just focus on regular unharmed people, and see if this plasmid DNA is integrating into the genomes of any of their stem cells. It leaves a calling card that is there. One of the reasons why I'm focusing on this is because it's kind of different from a lot of the other imagined harms where you can't really prove it. Really applied that regulatory limit to this new kind of vaccine where everything is encapsulated in this lipid nanoparticle. It's basically packaged in a synthetic virus able to dump its contents into a cell. So I'm thinking Hanlon's razor here. Okay, I don't think there was anything nefarious here. I think it was just kind of a dumb oversight. And it's going to take, because the financial incentives are so great to just, sweep it under the rug, and the career incentives of people that approve this are going to be, there's nothing wrong here. It's going to take some encouragement to make people prove that it's okay. But I really believe this was an inappropriate application of an old school regulation to a new kind of vaccine. And who knows, maybe we'll check a bunch of people and we'll find out for sure that this is indeed not a problem. And that will do the public good if we prove that.
There was a new COVID vaccine and this one really works.
There's no evidence to that as far as I can tell.
I understand that, and that's why I'm asking this question. Is there some way you could get a hold of one of those and do the same study that you did on these vials to make sure that we're not using that DNA protein or whatever it is, that the DNA that we don't need to be injected into these to our constituents?
I would like to do that, and I will not get it unless I get a batch and find out that it's free of DNA, and then I'll take it myself. But I don't have any way of compelling that to happen.
So it was just basically a way to save money by doing it in such volume that way without then taking it back out later on?
I think nobody thought about it. I think it was reasonable to use the E. coli to blow up the plasmid to make the stuff, and then the pieces of the DNA are of a very uniform and small size. That's evidence that they took efforts to try to chop it up. And
then they knew about it.
Yeah, they knew it, and they took efforts to chop it up. They just didn't get it all out.
But having said that,
I guess that they just didn't think about the hazard for genome modification, because it's not all that expensive to add another process to get it out.
Well, it seems to me that our people ought to be able to look at that, Mr. Chairman, and see whether or not it's got this DNA. And if it does, fine, tell everybody it's got the DNA and the problems associated therewith. And then you got informed consent.
Correct.
Okay, but without that, we don't have informed consent. I'm not really happy about that, Mr. Chairman, and do what I can to try to help.
Knowing what I know,
otherwise.
Knowing what I know now about this, I would still have recommended it to my elderly parents.
Okay.
But I probably would not have given it to my daughters.
People had this kind of vaccination. Do you know off the top of your head? Like the majority of people that took the vaccine have this type, because weren't there multiple types of vaccinations you could take?
The vast majority of people got either Pfizer or Moderna.
And we're talking about the Pfizer.
I'm talking about Pfizer. My colleagues have looked at Moderna, and it's in Moderna too. In the few that we've looked at, it's just not quite as high. I had a lot of experience with DHEC rolling out the saliva test. Okay, so we invented the saliva test, and then we had to deal with DHEC to try to get it rolled out for the state. And it appeared to me that they were just overrun. They weren't prepared for what this pandemic was. And it's no fault of theirs. I told people at the time that I felt like these were hobbits in the Shire that were accustomed to take care of small problems, and now all of a sudden we're in the War of the Ring, and there are orcs at the gate, and we're expecting them to deal with this tremendous challenge, and that's not who we put there. And that's not their fault. I mean, it's just we were not prepared to handle something of this magnitude. So some amount of grace, I think, is appropriate.
Findings in a journal.
No and no. They're not. As of now, they are interesting and concerning, but they don't rise to the level of a peer-reviewed publication. The most likely, the best possible outcome would be that I would check a bunch of people, find out it never integrated, and this is not a problem, and then it will never be published because papers don't publish negative results. Kind of the worst outcome is I can check a bunch of people and I find, it integrated and it's causing these horrible things. It takes about 3 hours to check a vial of vaccine to see if it's got this in it. About $100 of reagents. And I'm not going to get it unless I find a vial that I can check ahead of time and make sure it's not there. And if it is there, I'll take a pass on it.
Thank you. Dr.